Thank you, everyone, and welcome to Room C. The topic in here this morning is climate and carbon. I failed to mention earlier that, unfortunately, one of the other moderators, Rafiq Islam, many of you know, not unfortunately, he's actually doing some work this week in Johannesburg, South Africa, with the U.S. State Department. So, unfortunately, he wasn't able to make it this week, and I just wanted to acknowledge his contributions and work on this session as well. So next, we're going to turn it over to Sean Lucas, who's with Kentucky State University, background really in soil sciences and plant health, the specialty crop, hemp, right, and organic production. And he's going to talk about Active C as a measure of soil organic carbon sequestration this morning. So thank you. Thanks for being here.
Thank you, Aaron. It's good to be up here. It's been about 5 years since I've been to the Conservation Tillage Conference, and Rafiq Islam is a good friend of mine, and he asked me to come in and speak again this year. I want to say it's hard to follow up Warren Dick. He was a mentor for me when I was at Ohio State for a couple of years working on a PhD. It's kind of like Justin Bieber following up Led Zeppelin, and I don't even dance, right? So thank you, Warren. I said it before, I'll say it again publicly, for all the mentorship. Just a person I've tried to model my career after. I coordinate the organic agriculture research program at Kentucky State, and I dabble with a little bit of industrial hemp as well. But for the last 20 years, I've really been thinking about carbon a lot. Everything I do in organic ag, I do from the lens of soil management. How can we put carbon in the soil? How can we best manage the soil to keep healthy nutrient cycling, healthy microbial populations, all the things that Warren and Dave Brandt talked about earlier. This is a picture of our farm in Frankfort. So Kentucky State University is located in Frankfort. We've got a 300-acre farm. 16 of it is under my control and certified organic, and so we use a lot of cover crops. That's a rye vetch cover crop. I don't know if it's as nice as Dave Brandt's, but we like what we're doing. Going forward, one of the things that we're dabbling with is organic no-till. So I was really impressed to see Dave's 2017 results where he didn't use any herbicide, and so it's possible. And I cribbed some notes when he was talking about Rick Clark earlier. But what I'm really here to talk about is active carbon, labile carbon, you might hear it referred to as a measure of soil carbon sequestration. Oops, nope. There we go. I like to think about soil carbon through the lens of soil health. You know, I in my introduction here, I coordinate the organic agriculture program. Everything I do is through the lens of promoting a healthy soil ecosystem. And for me, that means putting as much carbon in my soil and maintaining that carbon as well as I can. And again, in organic, we haven't mastered the no-till angle yet. And so for us, we have to do a lot of tillage for weed control and seedbed preparation. And so I'm always trying to get whatever I can get in my soil in terms of carbon inputs. I like this quote from Franklin Roosevelt: "A nation that destroys its soils destroys itself." And we heard Dave Brandt earlier talk about just a recent silt storm. What do you say, 3 feet of silt on top of vehicles in the Southwest? This is the Dust Bowl that he was referring to. Not recently, but he also mentioned the Dust Bowls back in the 1930s, right? And we learned from that. If we're not keeping our soil armored, keeping our soil covered, we're going to incur damage. Dust bowls or soil crusts, which make it hard for our plants to germinate and peek up through and do the photosynthesis that they're supposed to do. Erosion, right? We're taking our best soil, it's leaving the landscape, and ended up really somewhere where we don't want it to end up. You know, I'm in Frankfort, Kentucky, and I always talk to our students in class, you know, if we have erosion in Frankfort, where does it go? Well, it goes to the Kentucky River. Well, where's that go? It goes to the Ohio River. Where's that go? Well, down the Mississippi. So potentially there's topsoil from Kentucky ending up in the Gulf of Mexico, right? And that's not where we want our topsoil. We want to keep it on our farms. Warren touched on this earlier. And again, I'm thinking of carbon through the lens of soil health and also through the lens of, look, I'm a dad. I'm worried about climate change. I got 2 little kids, and I want to leave a great environment for them after I'm gone. And so I think about it from the perspective of keeping carbon out of the atmosphere that we don't want there and keeping it in the soil where it has a benefit. And this idea of soil health has evolved over the last 20 years or so. We've known about it for a long time. I'm at an 1890 land-grant historically black college. George Washington Carver was talking about soil health back in the late 1800s, early 1900s. So we've known what soil health was for a long time, but we've really started to pound on it the last 2 or 3 decades and realize the importance of it. Before the 2000s, we were talking about soil quality. You know, that's the ability of a soil to carry out certain functions. Soil health is essentially the same thing, but we realize the importance of the biology in the system. As Warren mentioned earlier, a civil engineer might have a different idea of what a quality soil is compared to for civil engineering, for building a house, compared to a farmer looking to grow a crop. So soil quality, the idea depends on the use. Soil health really looks at the big picture. Oop, I'm trying to point, and I need to be clicking. Sometimes these terms are used a little bit interchangeably, right? But soil health in modern times has kind of taken more of a precedence. We're gaining an understanding of the importance of biology. We're treating our soils like an ecosystem. We know that we can monitor soil health. We can assess it and monitor it. It's not easy. There are challenges to understanding what the different things that we look at mean. And so I will get into that here in a minute. The goal, right, so that we can get more Dave Brandt-like farms and less farms like his poor neighbor, who's probably mad at him whenever he shows that picture, is to be able to monitor and assess soil health and then make recommendations and inform best management decisions. This is a nice cover crop. It's a winter pea, crimson clover, and you can see a little bit of rye coming up in there, a nice mixed cover crop. You're getting legumes for nitrogen input. You're getting a lot of carbon biomass from that rye. These are the types of management practice that we want to encourage that will help build soil carbon. And to know how we're doing in our management decision, we want to be able to monitor and track those carbon inputs. Soil organic matter is 58% carbon. For me, it's the key determinant of soil health, especially, again, thinking about managing organic systems where I can't go out and buy that silver bullet fertilizer. All of my nitrogen is coming from nutrient cycling from cover crops or from manure inputs, right? Organic materials breaking down and releasing nutrients. As we get more organic matter in the system, Warren showed a great picture earlier of a soil aggregate and a cross section. That's soil structure. That's what's holding our air. That's what's holding our water. That's what's allowing Dave Brandt's field to take 5 inches of rainfall in an hour and infiltrate it. And that soil organic matter is food. It's food for the microorganisms that live in the system. And those are the guys that are cycling those nutrients that our crops depend on. So again, for me, this is the key manageable component of an agro-ecosystem. Whether you're organic, whether you're conventional, and I hate that term conventional, right? I mean, we got to put labels on things so we can understand what we're talking about. Compared to organic, you know, we'd call Dave Brandt conventional, but he's by no means conventional. The stuff that he's doing is innovative and highly sustainable from my perspective. And I'm taking notes so I could steal ideas from him so I could get new ideas to research, right, when I go back to Kentucky and sit down behind my desk and start writing grant proposals. But we know that soil organic matter, super important in our agroecosystems. It responds to management. How we treat it is going to determine how well our soil performs going forward. Things like tillage, things like erosion, monoculture cropping systems, these are problems that will degrade soil organic matter, lose soil carbon. And again, soil organic matter is roughly 58% carbon. If I send my soil sample, I'm in Kentucky, so I send my samples over to University of Kentucky Regulatory Services. They do the soil organic matter test, and they just do a loss on ignition by combustion. And if I ask them, they'll give me both soil organic matter and soil carbon. And all they do to get the organic matter is they take the carbon number and they multiply it by, or divide it by 0.58. So that's a good rule of thumb. Soil organic matter is roughly about 58% carbon. That's what we're really talking about when we're trying to manage our inputs and monitor our inputs and the effectiveness of our inputs. We're really thinking about the carbon side of things. Soil health or soil carbon, you know, these are things, since we can manage it, we can improve it, or we can make it deteriorate. So the tillage, the erosion, the monoculture cropping, you might end up with a system with deterioration. If you start adding in cover crops or diversified crop rotations or manure inputs, you might be over here in the improvement zone. Soil organic matter wants to stabilize. It wants to find an equilibrium. It's going to depend on your temperature, your soil texture. Different factors are going to play a role in what that stabilized state looks like. But you can always go out of a stabilized state and into either deterioration or improvement, unless, of course, you're in that saturated zone, which we kind of shoot for, the pristine forest soil or the grassland soil that's undisturbed, right? It's going to be hard to add soil organic matter there. But generally speaking, agricultural soils, we're going to either be deteriorating or improving based on what kind of management we're doing. Assessing soil health is complicated. Carbon is the thing we generally look at to try and get a gauge about how healthy our ecosystem is, how healthy our soil is, our agroecosystem soil. We can't just measure crop yield and get an idea what our soil health is. Water quality, it'll give us an indication. You got water coming off your field like Dave Brandt showed in the pictures earlier, and that's, you know, that looked like he was managing his water quality and his ecosystem pretty well. When we think about soil health in the big picture, we need to think about how that soil carries out all the functions that we want it to carry out. Is it infiltrating? Is it keeping our water clean as it goes into our groundwater? Is it producing high quality, high yielding crops in a sustainable landscape, right? These are all the things that go into how we should think about assessing soil health. But that's a big bucket of stuff that we got to measure. And so if we want to get a quicker and dirtier estimate, then that's when we start getting into measuring soil carbon and soil carbon parameters. Warren talked about some of this earlier, so I don't want to belabor it, but this is important stuff. I love being a soil scientist. It's not a pure science. You've got to think about lots of different things. Biology, physics, chemistry, right? You've got to be able to process all those things and how they work together. And soil organic matter is involved in all of them. Decomposition, residue decomposition, nutrient cycling. Those are all biological properties that soil organic carbon influences. It's the food for the microorganisms. Physical properties, infiltration, water holding capacity, that's all determined by soil structure. Generally speaking, the more organic matter you have, the better your soil structure is going to be. The more porosity your soil is going to have, the better your roots will be able to penetrate that soil. And then chemistry. You get into this soil organic matter that's highly decomposed and has been there for a long time. Warren had the great bank analogy, which I'm going to steal, by the way. I'm going to steal that for a class at some point soon. And he talked about the long-term retirement account that doesn't really change, right? That type of organic matter that doesn't really change is giving you lots of chemistry, cation exchange capacity, which holds your nutrients, holds toxins that we don't want to get into our water systems. And it helps us buffer our pH. That can be a challenge if you've got a high clay, high CEC soil. You got to add a little more lime. But this soil organic matter does play a role in all these different processes. When I think about soil organic matter, I really love the sort of the description that Fred Magdoff came up with in Building Soils for Better Crops. Anybody heard of Building Soils for Better Crops? Yeah, it's an older book, but it's a book that you can get for free from the SARE website, I believe. And it puts a lot of the things that we're thinking about here today in everyday speak terms, right? Not science speak terms. And so Fred Magdoff, he came up with this sort of characterizations of different types of soil organic matter. And he talked about the living and the dead and the very dead. And he said, well, the living is the stuff that is living in our soil, plant roots, small animals, insects, arachnids, mites, right? Those are arachnids. Nematodes. Warren broke it down earlier and he gave specific numbers on all these critters. Bacteria, fungi, right? I love this picture. I show this in a lot of my classes at Kentucky State. This is one of my favorite soil science pictures of all time. You know, you can look at this and you can see the plant root. This guy is the root. That's the tip of a root, right? That's pretty fine. You know, that's the tip of a root. It's living. We think of that as part of the living organic biomass in the soil. When this root dies, it's going to turn into soil organic matter, soil carbon. This looks like a root, but that's not a root. That's fungus. And that's moving through the soil and holding soil particles together and helping create that soil structure that is so beneficial for air movement and water holding capacity. But I like to point out these little wispy things, right? Those are mycorrhizal fungi. They live in the root. They live on the root. And they get into little places in the soil where this root is too big to get. And they help bring nutrients and water to that root. And as you build organic matter, you're encouraging more biology, right? And more biomass of these types of organisms. Again, this mycorrhiza, it's going to bring phosphorus and water from pockets in the soil that root can't get into. That root benefits. That plant benefits from that. What's the fungus get? Well, the soil is a pretty harsh environment. Lots of like chemical warfare going on between microorganisms, right? They get a nice, safe place to live in the plant root. They get to freeload on carbohydrates that the plant is making through photosynthesis, right? So they get some food, but they help the plant out. They bring in water and nutrients. So this is part of the living organic matter. Let me think about the dead organic matter. This is the recently deposited stuff that serves as microorganism food, and this is where a lot of our nutrient cycling is taking place. This is the stuff that's, you know, last year's crop residues or the cover crop that you just rolled down. I had the privilege when I was at Ohio State in 2004 or 2005, I believe it was, to visit the triplet Van Doren plots with Dr. Dick. And they look similar to these plots. These are the Blevins Fry plots at University of Kentucky. And one of the things that always struck me about both the triplet Van Doren plots and the Blevins Fry plots, what these are, a comparison of no-till versus plow-till. In very simplified terms, that's what they are. And I noticed the same thing at both sites. This is the no-till plot. This is the plowed plot. And it's kind of hard to see from the perspective of this picture, but that no-till plot is about 6 inches taller than the plow plot. And that's 40-some years, or getting close to 50 years now, of building soil carbon, building soil organic matter, fluffing that soil up over time and decreasing the bulk density. All the things you want from organic matter, it's kind of represented in that little elevation change to me. We talked a little bit about the very dead. This is, humic substances, highly decomposed organic matter, super dark in color. Anybody in here compost? Compost is essentially humus. If you're doing a good job with your compost pile, you're finishing the compost. That stuff that you get at the very end that's really nice and dark brown, that's what humus is. That's what gives our soils the darker color. I hope Dave Brandt gets to his 49-inch depth with his dark brown color accumulation. This is the stuff that kind of provides that bulk density, the fluffiness that holds water, provides air movement, and facilitates soil structure. And as I mentioned, right, so I'm focused on soil health here, but we need to think about carbon in terms of keeping it out of the atmosphere. When I started as a graduate student at University of Maryland in 1999, and I was learning about climate change, and we were learning the numbers, carbon in the atmosphere was 380 parts per million. Now I'm at Kentucky State, and I teach classes at Kentucky State, and I'm telling my students, what the parts per million CO2 in the atmosphere is, and it's only been, 23 years. It's not that long. Anybody know what it is right now? 418 parts per million. Right? But historically, if we go back, the historical records have it at around 280. So back in the 1800s, it was 280. By the time I was a graduate student at University of Maryland, it was 380. And now in the last 20 years, we're up to 417, right? That's a pretty big increase. We want to keep the carbon out of the atmosphere. Agriculture is not going to solve the whole problem, but we can play a role in slowing it down. Our plants are our great carbon sinks, right? They take the carbon out of the atmosphere, turn it into photosynthates, and eventually those plants die and decompose and become soil organic matter. Some of that organic matter decomposes and turns back into carbon dioxide in the atmosphere, but we're catching some of it. And if we're managing our soils well, we can catch more than if we're not managing our soils well, which is kind of getting me to the point of my talk today, right? We need to be able to monitor this, and we need to be able to track it, and we need to be able to help producers inform their decisions. Producers know what soil health is. They've known it for a while. They can walk out and tell by the way a soil feels. Producers are, I look at Dave Brandt, he's ahead of where scientists are. Like I said, I'm stealing ideas from him so I can start thinking about, can I put an experiment together and start putting hard numbers to the things that Dave Brandt sees or the Steve Groffs of the world see and know is working for them, but can we start putting hard numbers? That's my job. Farmers are already doing a lot of, innovative farmers are already doing a lot of these things. They know what soil health is. They can go out, feel the soil. They know it's got good soil tilth. You smell the soil. It's got a good earthy smell to it because of some of the byproducts, the microorganisms. Which one of these is the healthier soil? Left or right? Why? Darker, more root mass, and you can kind of tell, it's not the best picture in the world, but you can tell that's got better structure. You could see the root penetration, right? And my old mentor at University of Maryland and one of my friends and colleagues when I was a graduate student there, Joe Groover, and Ray Weil was my mentor. They did a study, and they found that all of the farmer perceptions of soil health were actually related to soil carbon parameters as opposed to mineral parameters, right? And I think we know that now, right? We have a good sense of that now. That was almost 2 decades old, right? But we kind of know this. So farmers know what's going on. If we're thinking about how do we monitor soil health, how do we test soil health, assess it, some things are better than others to measure. You know, your pH is low. You can go out and you know, put some lime on your field or have someone come spread some lime on your field, and you could change your pH pretty fast. That's going to help your soil production, your crop production. Your fertility is low. You can add nitrogen. You can add phosphorus, potassium. But those are not things that we can measure that are really going to give us a picture of how our overall soil health, and again, I'm thinking of soil carbon through the lens of soil health, these aren't going to give us a picture of how that's changing. You know, and over here on the right, yeah, you know, I mean, if you don't manage your soil, you can change your soil depth, right? You're going to have some erosion, and you're going to change the depth of your topsoil. But assuming you're doing some management so that you don't have that problem, you know, you can't really change your soil depth. You could come in and have some land graders come in. That's a pretty expensive process, right? So you can't really change the topography or relief. I suppose you could truck in some sand if you had a heavy clay soil, but again, that's not very efficient, right? So these are things that are inherent soil properties, the factors of soil formation that you can't really change. What we're after are things that change over a few years to maybe up to a decade. Your soil structure, we can get at that by measuring aggregation. You can take microbial measurements, right? And you can monitor the level of microbial activity or community composition, which gets complicated because you have a lot of lab work involved in that, although the Solvita test has kind of changed the game on some of this. But what I like to think about are the labile carbon and total carbon pools. And the labile pool is associated with that living biomass, living organic matter pool that I discussed earlier. And that total soil carbon is more related to the humus and the dead stuff that's in there. So this labile carbon is a tool that has been developed over the last 20 years or so that gives us a little snapshot of how our management is impacting our soil. It changes a little quicker than the total carbon. Total carbon, you're looking at maybe decades to measure real changes. Although I've seen it in shorter than decades. You know, if you're really pumping in the organic material, you can see changes in a couple of years. But the labile carbon is a little more sensitive. You might pick up those changes a little earlier with the labile carbon. Yes, sir. Exactly the same thing, and I'm going to talk about that here. You know exactly where I'm going. You know, I mean, so soil health assessment, there's limited options out there. Some of our commercial labs are offering some options, but they can be expensive. Ohio State used to have a soil health testing lab, but you guys still offer services to the public? I think since Steve Coleman left, that is kind of, I don't want to say mothballed, but temporarily maybe that situation has changed. But, you know, it's hard to really get a picture in using laboratory methods on how our soil health is improving without doing some cumbersome work, right? In Kentucky, Waters Ag will offer, and I don't know if you guys, I tried to snoop around last night on the internet, and I couldn't find a comparable commercial ag lab up here that does this kind of work. But they have, you know, Waters Ag in Kentucky, they've got the basic soil test, which is just kind of like sending your soil sample to your state soil testing lab. And you can request a couple other things: nitrate, nitrogen, ammonium, nitrogen. They'll give you the Solvita test and the CO2 burst test, the SLAN test, which is a measurement of available nitrogen. But they throw active carbon in this mix. And they do some aggregate stability, and then they'll give you total carbon. And I'm going to shift now from talking about sort of soil health into now getting into permanganate oxidizable carbon for the last little bit of this discussion. If you're interested in soil health, even though the testing component of it has been kind of mothballed, there's some great information on soil health overall at soilhealth.osu.edu. And NRCS has a excellent primer on soil health at their webpage. So permanganate oxidizable carbon is a test that was developed really back in the 90s by a couple of Australian scientists. Blair was one of them. And my former advisor at University of Maryland kind of refined this test. The Blair method really was more of a measure of total carbon. Permanganate works by oxidizing carbon. And the Blair method, they used a higher concentration, and they were oxidizing just about everything. And so while kind of toned it down, and instead of using 0.333 molar potassium permanganate, they decided to use 0.2 molar, which only oxidizes, instead of getting all that carbon, you're only oxidizing the most labile carbon compounds in theory. In theory. This is not a perfect test. In theory, you're only oxidizing the most labile compounds. But it's a good test that'll enable us to do field testing. This is a field testing kit. That was me back when I wasn't gray-haired. I was at University of Maryland in that picture with a field kit doing a demonstration. And what you're measuring is the color change. And we're looking at two different soils in this picture. One of them had higher organic matter. One of them had lower organic matter. And the one with higher organic matter gives you this tube with the paler pink color. You're simply just measuring a color change. The more organic matter, the more your dark purple solution goes from dark purple to a paler purple or even a light pink. This test is sensitive to management. Steve Coleman did some great work with it here at Ohio State and really correlated it to basal respiration, microbial biomass, not just in one study, but in a big meta-analysis across multiple studies. Correlated also with particulate organic matter carbon, if you're familiar with that. But these are all sort of harder to assess, laboratory intensive methods that this permanganate method, which can be used in the field or in the lab, can get at much simpler, in a much simpler way. What are we doing? We're basically mixing 2.5 grams of soil with a permanganate solution in calcium chloride. We shake it for 2 minutes. The calcium chloride causes the clay particles and the soil particles to flocculate, which is just a fancy word for stick together and settle out. So we shake it. Calcium chloride causes it to settle. You don't need a centrifuge, right? You can just let it sit for 10 minutes, and your soil settles, and you're left with this supernatant, the upper part, the solution, which is what you're actually testing the color change in. And you measure that color change at 550 nanometers on a spectrophotometer. And then there's a formula that we use to calculate the labile carbon. But that's it. It's a pretty simple test. We'll go over some of the utility and caveats here in a little bit, but we know this test is useful because it's correlated very strongly with total carbon. So you can't necessarily see changes in total carbon in the first couple years of changing management, but you might be able to pick it up with this labile carbon test, and the labile carbon test is highly correlated with total soil carbon. So now we could start thinking about, all right, if I'm seeing changes early on, long term, maybe I can expect more and see observable changes in my total carbon. Maybe I can identify soils that are low in carbon now out in the field in a quick and dirty field test, and we could see soil candidates where improved soil organic matter management would actually put more carbon into the soil, benefit our crops, and keep carbon out of the atmosphere. We're talking about carbon markets now. We're talking about climate smart agriculture. We need a way to assess that easily, right? If we want to see where fields that would benefit from climate smart agriculture are, we need a test to identify them. And instead of having to send it to a testing lab and wait, for several months, maybe you can get boots on the ground with a test like this and get a quick read on it and then start making decisions. This is data from some of the work I did at Maryland in the early 2000s. And I went out and I identified fields at 4 different farms. Some of the fields had been tilled long term. Organic matter had been beat to heck. Others had recently come out of sod, and so they had higher organic matter levels. And what we did in these fields, they were side by side, they were the same soil type, 4 different farms. We put a cover crop treatment on. And this was, early 2000s. It wasn't a multi-species cover crop. It wasn't even rye vetch. It was just a heavy rye cover crop. And I was measuring different soil functional characteristics as well as crop yield characteristics. And what we saw was when you were putting a cover crop on in the soils that had low permanganate oxidizable carbon, when we added the cover crop to it, we saw more of a response, a change in yield compared to where we had no cover crop. We saw more of a response where the permanganate oxidizable carbon was lower. It kind of makes sense, right? I mean, if you already have higher soil organic matter and you're doing improved soil organic matter management, you wouldn't expect maybe as much change in the crop response. We saw higher functionality, more nutrient cycling, more mineralizable nitrogen, where permanganate oxidizable carbon was higher. And so this is getting towards, the overall functioning of the ecosystem. And again, this is just trying to reinforce the idea that this test, it's not a perfect test, but it's a pretty good quick and dirty field test that will indicate perhaps fields that can benefit from soil organic matter management. This is more recent work I did. Again, this stuff is sensitive to management. I took hemp and I had, anybody here grow hemp? For fiber? CBD. This was grown for fiber, and we were interested in the retting process. And if you know anything about fiber production, and y'all used to have a fair bit of hemp production here in Ohio before Prohibition, and Kentucky, of course, we're pretty well known for the work that was done with hemp in the 1800s up until shortly after World War II. And now that we've had a resurgence in interest in hemp, the CBD boom came and went, but people are still interested in fiber. People are still interested in grain. People are still interested in CBD too, but you kind of got to position yourself well in that game if you're going to succeed. If you are growing hemp for fiber, the traditional way to process the stalks so that the fiber can be easily extracted is to put the stalks through a process known as retting. Retting is a process by which you put the stalks on the soil surface. You allow them to decompose for 3 weeks, a month, a little over a month, depending on the weather conditions. You got more moisture, you might want to take them out sooner. You got a dry fall, you can leave them out longer. What we did was we set up a study where we had control plots where we didn't grow any hemp and we didn't have any fiber hemp retting on the soil surface. Then we had plots where we grew hemp, but we didn't allow the hemp to ret on the soil surface. Then we had plots where we put a sort of a half dose of hemp retting on the soil surface. I think it was about 2,000 kilograms per hectare of stalks on the soil surface. And then this high density HDR is about 5,000 kilograms per hectare of hemp on the soil surface. And we were really interested in, this retting process. We're having soil residues on the soil surface, or crop residues on the soil surface. How is that affecting our carbon? How is that affecting the microbial community, which is data, I have that data, but I'm not presenting it here. I wanted to focus more on the carbon side of things. But this is after a month of retting. 3 seasons of retting. 3 seasons of 1 month of retting. So this is the cumulative data after 3 growing seasons, where in each season there was retting involved at the end of the season. Where we had higher residues for manganate oxidizable carbon was showing us that we had significantly increased that labile pool of soil carbon. Interestingly, we were also picking it up in total carbon. So again, it's that relationship between this permanganate method, which is a pretty easy to use field method or lab method, or this total carbon method, which involves sending your sample in and doing a pretty intensive combustion process where you need a lot of equipment. One of the things I'm working on at Kentucky State is integrated crop livestock systems. We have a goat herd at the university. And I'm at an 1890 land grant. We deal with a lot of small scale producers, underrepresented and under-resourced producers. And a lot of them are interested. They don't have the land to run cows, right? But they might be able to put goats on the landscape. And so I got there in 2016 at Kentucky State, and I saw the goat herd, and I had been thinking about doing an integrated crop livestock experiment for years, and I talked to our goat guy, and I said, hey, can we do this? He said, yeah. So we started an experiment where we had a 5-year crop rotation, organic corn, organic soybean, followed by 3 years of pasture. That's the 5-year rotation. So you got corn, soybean, pasture year 1, pasture year 2, pasture year 3. And we also, in this study, put in plots where we were just cropping every year. Corn, soybean, corn, soybean, corn, soybean, corn, soybean. And we had a separate plot that was just always in pasture, never disturbed. So here's some of our goats. You can see the goats on this plot, and I try to follow the take 50, leave 50 pasture rule, where when they graze it down about 50%, and goats are not natural grazers, right? They're browse animals. They don't like to be forced to put their head down. That's essentially what we're doing, though. So we work with Welter's Seed and Honey out in Iowa, and they put together a really nice custom mix with some forbs and some tasty things for goats that, even though we were forcing them to eat the grasses, they were also getting some things they liked. They did a pretty good job, and interestingly enough, depending on the time of year we put them in, earlier in the season they would seem to go for the Johnson grass as well, which I want to get the Johnson grass out of my plots. Later in the season they didn't seem to like it as much, and that's when I got my seed heads showing up on my Johnson grass. And again, I'm organic, so I can't spray it down. I was hoping the goats would do a little better with that, but we're working on it. This is a plot that was just recently grazed. And this is the perennial pasture that's never grazed. And you can see the corn plot to the left. We've got each treatment of that 5-year rotation represented in every year of the study. So every summer I have corn, soybean, pasture 1, pasture 2, pasture 3 sitting next to each other in the field, right? Along with the perennial pasture and the continuously cropped sites. Here's what the goats are leaving behind, right? That's organic matter to me. They're chewing up my grasses, my forage, and since I'm trying to promote or trying to practice take 50, leave 50, hopefully we're stimulating some good root growth, right? Through a healthy graze, not a detrimental graze. But they're leaving little nuggets of nutrients behind, nutrients and organic matter. Plus, I'm not disturbing that. For 3 years, I'm not disturbing that pasture. So what's that get us? Well, this is using permanganate oxidizable carbon. Again, this stuff is sensitive to changes in soil management within 3 or 4 years. This is data that came after 4 years. We didn't see significant differences, although we saw this same trend showing up in our data in the first 2 to 3 years of the study. But by the 4th year of the study, these differences were significant. Well, what differences are we looking at? This is the continuously cropped corn, soybean every year, right? This is the perennial pasture. This CS1 was the corn phase of our rotation. CS2 was the soybean phase, and then we've got 3 years of pasture, right? And now the caveat here is this is the first 5 years of this study, and it had been in pretty intensive agricultural production before we initiated the study. So these organic matter levels were already kind of low. And so what we're seeing is when we're in that first year of cropping, the organic matter levels are similar to continuous cropping. And even though there's a bump here in the soybean year, I don't know that that's a true increase in soil organic carbon. It kind of looks like it is, but that's another year of tillage, and so I'm a little suspicious of whether that's really an increase. It's not statistically different. I think it just might be some slight variation in the data gave us a look that kind of fits with the rest of this. But what I want to point out is when we go back into pasture, look at that organic matter accumulation. We're approaching, by the 3rd year of pasture, we're approaching the organic matter, permanganate oxidizable carbon organic matter, that we have in an undisturbed pasture. And so what that tells me is someone who's interested, in soil management, especially in organic systems, is that, yeah, I want to shift to organic no-till at some point, but we don't have that all dialed in yet. Every region is different, right? Glyphosate works the same in Maine as it does in Kentucky, as it does in California, but Maine and Kentucky and California all have different ecosystems, and so if you're going to do organic no-till, you've got to figure out what that system is going to look like in your particular ecosystem. And we haven't quite dialed that in yet. But I still know that I can go ahead and do 2 years of tillage to produce organic corn or organic soybean, and if I follow it up with animals on pasture for 3 years, I'm almost getting back to an undisturbed situation with my organic matter. And those goats, that's diversification, right? A lot of people are interested in goat meat, particularly around different ethnic holidays. People of Latino heritage, people of Islamic heritage, lots of their holidays rotate around a feast that involves goats. So there is a niche opportunity to provide meat for those communities. That's diversification. Same study, and this graph looks funky. I worked on this last night, and you can see it's got some different color tones in it. And the reason it has different color tones is not because I'm cheating and lying on my data. I just did Microsoft Paint, and I cut the picture apart and put it so that it was in the same order as the previous picture. This I stole from my graduate student. And I didn't have time to run the numbers on the same program that I use to make this figure, right? But I wanted the x-axis to look the same. And so I basically just cut and pasted the bars so that CMP, CS1, CS2, all the way over to PP were the same. But whatever my graduate student used to generate this figure, it had different color tones in it. So that's why we see the kind of funky coloration scheme here. But this is aggregates. And so what we're seeing with permanganate oxidizable carbon is following in what's going on with soil aggregates. We're rebuilding our soil structure. We know that with tillage, we're coming in, we're destroying that structure, we are introducing oxygen, we are opening up those soil pore spaces that hold that soil organic matter and allowing microbes to attack that organic matter and giving them the oxygen they need to do it fast, right? And so in this cropping phase of the cycle, we're seeing a decrease in soil structure. But as we move back into pasture with goats on the landscape, we're not quite back up to perennial pasture status, but we're getting there. So I guess to summarize, I mean, this is a pretty useful tool. We can use it in a field kit. We know it responds well to management. It can help us predict soils where we may be able to implement improved organic matter management practices to sequester carbon intentionally. There are caveats. This is not a perfect test. You can't just get your 400 milligrams per kilogram reading of permanganate oxidizable carbon and say, well, that's good. That number is going to be different for different soils, depending on your climate, depending on the texture of your soil, and some other conditions. And so it's best used on a relative basis. You might want to get a baseline number and then work from that baseline to see how things are improving. We also know that there are some methodological inconsistencies with this method. Not inconsistencies that take away the usefulness of it, but you got to understand what these inconsistencies are when you're using it. And so, one of the things we found is that you can use 5 grams of soil. The original method called for 5 grams of soil. But if you have a super high organic matter soil, all the color would be gone from your permanganate solution. And so then your reading at the end didn't really give you any meaningful data. You're just using up all the permanganate. So we scaled it back to 2.5 grams, and that, even with our high organic matter soils, was giving us good readings. But if you plot the data, the 2.5 gram and the 5 gram, there should be they should be parallel, but they weren't necessarily parallel lines when you looked at the data. So there's some methodological inconsistencies. Soil texture, higher clay soils, you may get the texture affects the way that the permanganate is able to access that organic matter. And soil sieving can also slightly confound the method. However, it doesn't confound it to the point where you can't get some useful information of it. Even with all of these things, you can still use this on a relative basis and track your progress or identify things in the field where you think things could be improved. So what do we need? What do we need? Well, obviously we're here and we're talking about this stuff. We need people to adopt more soil carbon building management practices. I think we're in a good group of people here to think about that. But we need to go beyond those of us here. And, Dave Brandt needs to keep working on his neighbor, and the rest of us need to go out and have good conversations on how we should be building our soil to keep the carbon in the soil, to do all the good things it does in the soil, and to keep it out of the atmosphere where it causes trouble. We need outreach and extension on soil health and on soil carbon. I think most of us here are probably fairly well informed, but again, I drove in here and I was astounded by just looking out my window last night at about 4 o'clock as I was pulling in, and there's lots of fields that are tilled. You know, and we see the same thing in Kentucky. Kentucky and Ohio they're the birthplaces of no-till, and we still got fields that are being tilled that I'm assuming aren't organic, right? The ones down the road that I saw weren't organic. I'm giving myself an excuse because I can't use herbicide. We need valuations on carbon. Jim Horman and Rafiq Islam in an extension publication coming out of Ohio State, they valued 1% of soil organic matter was worth about $680 an acre in organic nutrients. I've told that number to other people and they kind of raised their eyebrows, but at least Horman and Islam took a shot at it. We need more studies like this to really figure out what the true value of carbon is as we're talking about carbon markets and carbon capture and farmers being incentivized to put carbon in their soils. We're starting to see incentivization. I'm part of 2 climate smart agriculture grants where some of the money from the grant is going to farmers to incentivize building soil carbon, and we're going to be tracking the progress in those grants. One of them is tracking 700 farms in Appalachia, in Kentucky, and in North Carolina, and we're going to be boots on the ground. Workshops talking about building soil carbon, and we're actually going to be monitoring over 5 years the work that those farmers are implementing. The carbon markets, I don't know. I mean, they're not clear to me, and I've been studying carbon for 20 years. So we need more evaluation. We really need to understand the worth of carbon. And permanganate is one tool, but we need more assessment technology. If we're going to really do this right, every tool we have is going to help. So that's what I brought today. Again, thank you all for having me up to CTC. And if we have time for a couple questions.